So welcome to today's tutorial. And our lesson for today, we are going to concentrate on translation. In our previous class, we looked at transcription and also the overview or introduction to gene expression. We mentioned that gene expression occurs via transcription and translation. So today we are going to be looking at translation. So our learning objectives will be for us to know and understand the genetic code, to understand the role of RNA and tRNA in translation, to also understand the process of translation itself, and also to know the post-translational modifications and their importance. So the outline for our lesson will have a review of the central dogma of molecular biology. We are going to look at the genetic code, and we're going to look at the role of RNA, and then we are going to look at the role of tRNA. Then we're going to dive into the process of translation, and then we're going to finally look at post-translational modifications and their importance. So having mentioned in the preamble that gene expression occurs via transcription and translation. So when we look at the central dogma of molecular biology, which links DNA replication, transcription, and also translation. So it gives us, or it tells us that the base sequence of DNA would determine the base sequence of messenger RNA, of which the base sequence of the messenger RNA is going to determine the amino acid sequence for the proteins that are going to be translated. Since we know that amino acids are the monomers that make up proteins. So today we are concentrating on translation, the process of translation. So our first part is us looking at the gene concept and the genetic code. So when we look at a gene, when we talk about a gene, we're simply talking about a sequence of DNA that is responsible for the production of a complete and functional protein. So a gene is simply a DNA sequence responsible for production of a complete and functional protein. And then we have what is called an anticodon or anticodon, which is simply a sequence of 3 nucleotides that is found in transfer RNA. And this sequence of the nucleotides is complementary to the codons, to the 3 nucleotide codons. And then we have a codon, which is simply a triplet of messenger RNA nucleotides. So the anticodon combines with the 3 nucleotide codon. So we have about, we have 64 possible codons, which constitute the universal genetic code, in which each codon specifies an amino acid in a polypeptide or in a protein. Or we have those that signal the start of protein synthesis or the termination of protein synthesis. So now let us look at the principles of the genetic code. So we're saying the genetic code is a set of rules by which a linear sequence of nucleotides specifies the linear sequence of a polypeptide. What we are saying that these specify how the nucleotide sequence of a messenger RNA is translated into the amino acid sequence of a polypeptide. So it links the relationship between the nucleotide sequence of messenger RNA and the amino acid sequence of the polypeptide. the nucleotide sequence is read as triplets, which are known as the codons. So basically, the genetic code uses the 4 different types of nucleotide, nitrogenous nucleotide bases. So we have adenine, guanine, uracil, and cytosine to code for 20 amino acids. So they generate the 20 amino acids in form of codes, and that's called the genetic code. So each codon represents an amino acid. However, when we look at the codes and the amino acids, we find that there are an excess of triplet codes than the required 20 amino acids. Therefore, the genetic code is termed as being degenerate or redundant, in that there are too many codes for few amino acids. So having analyzed the genetic code, and after the scientists cracked the genetic code, they realized that it had several series of important properties or characteristics, which we need to look at. So the first one is that it is a collection of codons, which we have already defined. So these codons, this collection of codons specify all the amino acids that are found in all the proteins. Then it is a triplet code, meaning that it has codons in which the nucleotides of messenger RNA are arranged as a linear sequence of triplets or 3 nucleotides, which are called the codons. So we are saying that each codon is 3 bases long and is a code for 1 amino acid. So singlet or doublet codes are not adequate to code for the 20 amino acids. Therefore, it was pointed out that the triplet code is the minimum that is required for the coding of amino acids. So using the same code, we are saying that when we look at the DNA code for each amino acid, it can be obtained by converting rRNA codons back into their complementary DNA triplets of bases according to the rules that are found in the genetic code. And then the other property is that genetic code is universal. It is universal. So by universal, we mean that with a few minor exceptions.
No. I'm trying his mouth.
So we have
so
we have a few minor exceptions, such as mitochondrial DNA, yeast, and some ancient bacteria.
To listen, guy. So after
it's being recorded.
So we are saying that with a few minor exceptions, such as mitochondrial DNA, yeast, and some ancient bacteria such as mycoplasma differ from the universal code. All codons specify the same amino acids in all organisms. So in short, all living organisms contain the same 20 common amino acids and the same 5 bases of adenine, guanosine, thymine, and cytosine, and uracil. And then the other property is that the genetic code is degenerate or redundant. So when we say redundant, we are saying that more than one codon may specify the same amino acid. So a given amino acid may be coded for by more than one codon. So a given amino acid may be coded for by more than one codon. So when you look at the amino acids, all amino acids except for methionine and tryptophan, all amino acids except methionine and tryptophan are coded by more than one codon. So when you look at methionine is coded by one codon. And tryptophan is also coded by one codon. And then we have other examples such as arginine, serine, and leucine, which are coded by 6 codons. So this is what it means by the genetic code being degenerate or redundant. And then the code degeneracy is of basically 2 types. So we have what is called partial degeneracy and complete degeneracy. So when we look at the partial degeneracy, in partial degeneracy, we find that the first 2 nucleotides are identical. So remember the codons are triplets. So in partial degeneracy, we find that the first 2 nucleotides are identical, but the third nucleotide of the degenerate codon differs. So for example, coding for leucine, we have CUU. We have CUU coding for leucine, and also CUC also codes for leucine. So look at CUU and CUC. We find that the first 2 nucleotides are the same. They are identical. They are all CU. And then the third nucleotide, we have in CU you have a U, and in CUC we have a C. So that is partial degeneracy. And then complete degeneracy occurs when any of the 4 bases can take third position and still code for the same amino acid. So in complete degeneracy, any of the 4 bases can take the third position and it will still code for the same amino acid. So for example, when you look at serine, the amino acid serine, is coded by, can be coded by CU, or rather it can be coded by UCU, UCC, UCA, and UCG. All these code for the amino acid serine. So when you look at UCU, we have uracil at the third position. When you look at UCC, we have cytosine at the third position. And when you look at UCA, we have adenine at the third position. And when you look at UCG, we have guanine at the third position. But they still code for the same amino acid serine. Therefore, any of the 4 bases have taken the third position, but they still code for the amino acid serine. So that is complete degeneracy. And then the amino acid is unambiguous or non-ambiguous. So by this, we mean that a single codon can never code for more than 1 amino acid. So a particular codon will always code for the same amino acid. Now, looking at degeneracy and non-ambiguous or unambiguous, we find that while the same amino acid can be coded by more than 1 codon, which we termed degenerate, the same codon will not code for 2 or more different amino acids, and that is called non-ambiguous. And then the other property is that the genetic code is non-overlapping. So by this, we mean that the adjacent codons will never overlap. So it means that the same letter is not used for 2 different codons. So in other words, we are saying no single base can take part in the formation of more than 1 codon. No single base will take part in the formation of more than 1 codon. That's what we mean. So each nucleotide is used only once. So this makes the genetic code to be continuous. And the other property is that the genetic code is commaless or comma-free. So by this, we mean that there are no breaks or markers to distinguish 1 codon from the next. So there's no signal to indicate the end of 1 codon and the beginning of another codon. There are no intermediary nucleotides between the codons. So it's just continuous. There are no breaks between the codons or the 3 nucleotide bases. And then the other property is that the code is conservative. So when several codons specify the same amino acid, the first two bases in those codons are almost always identical. And then the code has a polarity. The genetic code has a polarity. So the code is always read in a fixed direction. That's what we mean by saying it has a polarity. It is always read in a fixed direction. And this direction is in the 5' to 3' direction. So from this, it is apparent that if that code is read in the opposite direction of 3' to 5', it would specify two different proteins, since the codon would have reversed base sequence. So no wonder the code has a polarity and read in the 5' to 3' direction. The other property is that the genetic code has punctuation marks, which signal the start of translation or the end of translation. So we have of importance, very important, the start codon, which is methionine, which codes for methionine, and this is AUG. So the start codon is AUG, which also codes for methionine. And then we have the stop codons, which are also called the termination codons or the nonsense codons, which are UAA, UAG, and UGA. So the stop codons are UAA, UAG, and UGA. So when you look at the start codon AUG, it has a bifunctional, or it is bifunctional. It has two functions. One, it signals the start of translation. And then it also codes for methionine. So two functions, signals the start of translation and also coding for methionine. So these are very important. You need to know the start codon and the stop codon. So using the genetic code and the central dogma of molecular biology, we are able to predict the codons and amino acid sequences that are encoded by a particular DNA sequence. So if we are given a particular DNA sequence, we are able to predict what will be the sequence of the amino acid. We're able to predict the codons and also the amino acid sequence. Remember when you looked at transcription, we talked about the sense strand and the antisense strand. And we said that the antisense strand is the one which is transcribed. And from that antisense strand, we are able to predict the messenger RNA sequence that can be obtained. And furthermore, predict the codons and the amino acid sequence from the DNA sequence. And then we are also able to determine the set of messenger RNA and DNA sequences that would code for a particular sequence of amino acids. So sometimes we may have a sequence of amino acids given, and using that amino acid sequence of a polypeptide or a protein, we are able to work back so that we are able to determine the sequence in messenger RNA and DNA sequence. So the genetic code is quite important. It's important to us scientists. So this is simply showing the genetic code. And then we have the principle or the hypothesis of Warburg hypothesis. So when there is an interaction between the codons on messenger RNA and the anticodons which are found on transfer RNA. So the anticodons that are found on transfer RNA, they need to be the same or exact in the first two positions. But that does not need to be so in the third position. So we found that in the third position, there could be non-standard base pairing, which may occur between the third bases of the anticodon on the transfer RNA and the codons on the codon on the messenger RNA. So that degenerate third base is said to be in wobble position. So in short, we are saying that wobbling happens at the third position. So An example of a non-standard base pair that can happen is between guanine and uracil. So these are acceptable in the third position. And even if there is that non-standard base pair, that won't change the amino acid that the codon specifies. And then we're going to quickly look at the role of ribosomal RNA. So when we looked at nucleic acids, we had an overview of the RNAs, the 3 types of RNAs. So I'm going to have a review and also add some more information. So when you look at ribosomal RNA, it is used to synthesize ribosome when it associates itself with proteins. And the functions of the ribosomes is simply to hold in position the messenger RNA, transfer RNAs, and the enzymes that are involved during protein synthesis. So the ribosomes hold the messenger RNA, the adapter molecule, which is transfer RNA, and the enzymes involved in protein synthesis. So for the ribosome to function, they need to first bind to at least 2 charged transfer RNAs at once so that a peptide bond can be formed between their amino acids. So we are saying the ribosome has basically 4 binding sites. There's 4 binding sites. So one binding site is for messenger RNA. One binding site is for messenger RNA, and we have 3 binding sites for transfer RNA. So this implies that the ribosome will hold the messenger RNA template. It will also hold the amino acid transfer RNA molecules and the growing polypeptide chain in the correct orientation so that the genetic code can be read and also the next peptide bond is formed. The transfer RNA molecules attached to 3 binding sites or 3 depressions that are found on ribosomes. So we have the peptidyl site or the P site. So the P site or peptidyl site is given that name because the transfer RNA holding the growing polypeptide chain occupies the P site. So the transfer RNA that is holding the growing polypeptide chain occupies the P site, hence the name peptidyl. And then we have the amino acid site, which is the A site, which is named so because the amino acid transfer RNA that is delivering the next amino acid in the sequence has to bind to this location. And then we have the exit site or the E site or the M site. This is where transfer RNA that has delivered amino acid to the growing polypeptide chain will exit the ribosome. So the exit site simply binds the transfer RNA that has carried the previous amino acid. So it exits through the exit site. So this is simply showing the binding sites. So I have the E site, P site, and the A site. And this is the larger sub unit of the ribosome. And then we have the smaller subunit of the ribosome. When the large subunit and the smaller subunit bind together, there's a groove that is formed. And yes, fourth, the messenger RNA will pass through that groove between the 2 ribosomal units. So the functions of the ribosomes, they have both decoding and enzymatic functions. So the ribosome has both decoding and enzymatic functions. So these 2 functions of ribosomes involve decoding the transcribed message, and forming peptide bonds. So the decoding function resides primarily in the small subunit of the ribosome. And then the formation of peptide bonds requires the enzyme peptidiotransferase, which resides in the large subunit. So the two functions of ribosomes, we have the decoding function, which resides in the small subunit, and we have the enzyme peptidiotransferase, which resides in the large subunit and is involved in the formation of peptide bonds. Then quickly, the role of transfer RNA. So transfer RNA transports amino acids from the cytoplasm to the messenger RNA, where the amino acids are joined together into a polypeptide chain. So each amino acid has its own transfer RNA, which carries a triplet anticodon for the amino acid. So remember that each amino acid has its own transfer RNA. And remember, as we had mentioned earlier, the anticodon on transfer RNA binds to the codon on messenger RNA. So the anticodon is complementary to the specific codon in messenger RNA. And the 3' end of the acceptor arm of transfer RNA carries the sequence or ends in the sequence CAA 3', which is used to attach a specific amino acid. So the amino acid binding site is at the 3' end of the transfer RNA. So what happens is that the carboxyl group of the amino acid will be covalently bonded to the exposed 3' hydroxyl group of the terminal nucleotide that will leave the amino group of the amino acid free to participate in peptide bond formation. So this is the hydroxyl end. This is the 3' end, which is the amino acid accepting end. So the hydroxyl group of the amino acid will be bonded to this exposed 3' end of the transfer RNA. So transfer RNA has a bifunctional, it is a bifunctional molecule. It has two functions. It has to be able to interact with the messenger RNA at the anticodon loop, and it should also be able to interact with the amino acids at the acceptor stem or acceptor arm. So the acceptor stem, as we have already mentioned, ends is at the 3' end of the molecule and ends in the 5' CCA 3'. So we have the anticodon for interaction with messenger RNA, and we have the hydroxyl group at the 3' end for interaction or accepting amino acids. So this leads to the molecule having a bifunction or being bifunction. So there are certain properties that are essential to the function of the transfer RNA. So one, it has an anticodon, which has complementary binding sequence to the correct messenger RNA codon, as we have mentioned earlier. Then the transfer RNA is also recognized by enzymes called aminoacyl tRNA synthetase that add the correct amino acid. And then it has an attachment site for the specific amino acid, which is specified by the anticodon. The other property is that it is recognized by the ribosome. So these four functions are very essential to the function of transfer RNA in the process of translation. Okay, so now looking at the process of translation, it has main steps, about 6 main steps in the process of translation. So we have the amino acid activation and attachment to transfer RNA or the charging reaction. That's the first step. Then we have the binding of messenger RNA to the ribosome, which is the second step. The third step is polypeptide chain initiation. So basically, the first two steps will lead to the polypeptide chain initiation. And then we have polypeptide chain elongation. And then after that, we have chain termination. And then lastly, we have post-translational modifications. Just as doing transcription, we have post-transcriptional modifications. After translation is done, we have post-translational modifications. So now the first step is what is called the charging reaction. The charging reaction. So remember, we talked about enzymes called amino acid tRNA synthetases. So these amino acids must be able to recognize specific transfer RNA molecules, as well as also their corresponding amino acids. So the amino acid tRNA synthetase, they need to recognize specific tRNA molecules and also their corresponding amino acids. This reaction, which is characterized by this enzyme, is what is called the charging reaction. So in the first step of the reaction, the amino acid is activated. So it reacts with ATP or GTP to produce an intermediate with the carboxyl end of the amino acid attached to AMP. And then the two terminal phosphates are cleaved from ATP during this reaction. So this is the diagram simply showing this process of the charging reaction. So when you have the AMP that is produced, the amino acid, there will be an amino acid AMP or adenosine monophosphate complex that remains bound here, that remains bound to the enzyme. So it remains bound to the enzyme. And then the second step of the reaction will transfer the amino acids from AMP to the transfer RNA, which will produce a charged transfer RNA and AMP. And then this charged transfer RNA consists of a specific amino acid that is attached to the 3' acceptor stem of its RNA. Remember, each transfer RNA has a specific amino acid that it attaches to. So in summary, this is what happens. You have an amino acid plus ATP or GTP in the presence of amino acid transfer RNA synthetase, which leads to an activation of amino acid. So you have an activated amino acid plus adenosine monophosphate. And then we have the activated amino acid, which is activated for methionine in prokaryotes, plus the transfer RNA will lead to a formation of methionine tRNA, which is an amino acid transfer RNA. So each amino acid is activated by its own amino acid transfer RNA synthetase. So for example, if you have lysine, it will be activated by lysine transfer RNA synthetase. Okay.